Welcome to Breakthroughs in Brain Development Through the Lifespan, an informative presentation from experts in the UCLA Neuroscience Research Team who have been conducting research on different aspects of the brain. My name is Coretta Harris, and I currently serve as President of Women in Philanthropy, having graduated from UCLA in 1983 in the School of Engineering, and I'm happy to continue to support the School of Engineering philanthropically. Women in Philanthropy at UCLA celebrates and inspires women's giving across the UCLA campus and fosters women's participation on campus boards and volunteer leadership positions. The group celebrated its 25th anniversary in 2019, and it now boasts over 3,100 members who contribute time, talent, and treasure to every corner of the university. These extraordinary women leaders have roles on many of the advisory leadership groups at UCLA and also hold positions within the UCLA Foundation. To engage the UCLA community, we hold events throughout the year that showcase different areas or topics across campus. For these large-scale programs, we're focusing on webinars to allow UCLA supporters from across the country to participate as we showcase different aspects of UCLA's programs, faculty, research, and innovation. Today's presentation should be very enlightening and informative. Brain disease undoubtedly presents one of the greatest challenges to medical science in the 21st century. We can see from the strong attendance and the quantity and scope of questions that were submitted, there are a lot of uncertainties surrounding the brain and its inner working. Fortunately, researchers throughout UCLA are dedicated to understanding the mysteries of how the brain develops and works, and then for creating effective new therapies when it doesn't. Today, we have convened a panel of female neuroscientists from UCLA Health and the David Geffen School of Medicine at UCLA to share significant discoveries in their research on different aspects of the brain as it develops, functions, and ages. I'd like to introduce our moderator, Dr. Emily Marcus, who will introduce us to each of the panelists. Dr. Emily Marcus joined the David Geffen School of Medicine at UCLA in March 2018 as Executive Strategy Officer and Adjunct Professor, Biological Chemistry. As Executive Strategy Officer, her responsibilities are centered on envisioning the future of a diverse and inclusive academic medical center, including the foundational pillars of the medical education, research, clinical care, and community outreach. Dr. Marcus earned a Ph.D. in biology from Yale University and conducted postdoctoral training at the University of California, San Diego, and the Salk Institute. This all makes her the ideal person to facilitate the discussion with the panelists. The panelists will share more information about their research and experiences and then answer questions from Dr. Marcus. There will be time after their conversation for the panelists to answer questions from the audience, so please submit them through the Q&A function on the bottom of your Zoom screen. They will be reviewed and consolidated by staff, and Dr. Marcus will present them to our panelists for their response.
Thank you very much, Dr. Harris. Welcome, everybody. It's really a pleasure to be here. I think this is going to be an incredibly exciting event. I often say that I was born a neuroscientist, but if not, I certainly have been one for a very long time, longer than I care to admit. And before joining UCLA, I was editor-in-chief of one of the premier journals that publishes in the neurosciences. So over the course of my career, I really had a very an opportunity, sort of a unique view on an ability to watch the field of neuroscience develop in two key important ways. And one relates to the science of neuroscience. I think here in 2022 and looking out to the next 10 or 15 years, we are really at an inflection point of breakthroughs in the field. We have new technology, there's new questions we can ask. I think the science has really come along to the point where we're really optimistic about being able to do research that's going to directly impact human health. The second actually is in the engagement of women in the field of neuroscience. So when I started out my PhD work at Yale, I was one of very few female neuroscientists, and I can say for certain that back then there would be no way that we could have a panel like the one we have today of leading women neuroscientists, and certainly not all here at one university like UCLA. So it's a real pleasure to bring those two things, two passions of mine together in today's panel discussion. And I think you're going to learn some really new and exciting things by listening to the panels. So in this evening's discussion, we've got 5 women neuroscientists who will discuss their fascination with the brain, the specific research that they do, how it relates to human health, and the challenges facing women neuroscientists. So all 5 are rising superstars. And by rising superstars, I mean they have already made incredible contributions to the field, but they're not done yet. And they have a lot more. They're in the sort of accelerated part of their research paths, and they have a lot more that they're going to be contributing to the field of neuroscience. So the studies that you're going to hear about today, they span the lifespan, starting from including early brain development, Laura DeNardo, how we make decisions, and how the brain degenerates. So at this point, I'd like to introduce the 5 speakers in alphabetical order. Aparna Bhaduri, who's an assistant professor of biological chemistry, and Churchland, who's a professor of neurobiology, Laura DeNardo, who's an assistant professor of physiology, Yirong Peng, who's an assistant professor of ophthalmology, and Jessica Rexach, who's an assistant professor of neurology. So with that, I'd like to turn it over to our first presenter, Laura, and I turn it over to you.
Okay, thank you, Emily. It's lovely to be here. So I'll talk a little bit about what my lab does at UCLA and the path that I took to get here. So my neuroscience journey started when I was a freshman at UC Berkeley. I wasn't sure what I wanted to study, and I took a class called Brain, Mind, and Behavior that really piqued my interest. So I started working in a neurobiology lab on campus and just fell in love with the science, but also with the sense of community in the lab. I had really fantastic mentors there who encouraged me to do a PhD. So I decided to pursue a career in academic neuroscience. So I did my PhD at UC San Diego, and there I studied mechanisms that help the brain wire up properly during development. Then I went on to do a postdoc at Stanford, and I started studying a brain region called the prefrontal cortex. You can see in my background is lit up in green. This is a coronal section from the mouse brain, which is the model organism that I study. So the prefrontal cortex is part of the frontal lobe, and it's really important for cognitive functions like memory, decision-making, attention. And while I was a postdoc, I discovered a special prefrontal circuit that's really important for storage of long-term memories. So my research program at UCLA, I started a little over 3 years ago, brings together these different kinds of training that I had. And we study how the prefrontal cortex develops. So the prefrontal cortex undergoes a lot of structural and functional changes into early adulthood. And this makes it one of the last brain structures to reach full maturity. So we think this long development that the prefrontal cortex goes through may be really important to establish its mature cognitive functions, but it also can lead to some problems. So when you have a long period of time where things are changing in the brain, it opens up a window where a brain region may be sensitive to insults. So these may be genetic mutations or environmental insults like early life stress or environmental toxins. And for this reason, prefrontal dysfunction is linked to a huge number of neuropsychiatric disorders. So these include autism spectrum disorder, schizophrenia, anxiety and depression are most relevant to the work that I do. And many of these disorders tend to rise early in life, in childhood and adolescence. So we're really interested in understanding how prefrontal development enables these complex cognitive and behavioral functions to emerge. And we think that understanding this can help us understand how these processes are altered in disease. So we focused on a few behaviors in my lab. One of them is called threat avoidance. And we're particularly interested in this because an important part of development is learning how to adaptively respond to our sort of changing, ever-changing and uncertain environment. In order to survive, we have to learn how to find rewarding things like food or friends or mates while avoiding threats. And this process, this balance is something that relies really heavily on the prefrontal cortex, but we don't really understand how this occurs throughout development. So some of the studies in my lab have been focusing on adolescence, which is a time when a lot of psychiatric symptoms emerge. And in healthy animals, and people, it's also a time where it's really important to explore so that we can establish independence from caregivers. So we can model this threat avoidance in adolescent and adult mice in the lab. And we find that adult mice are fairly risk averse. They work hard to avoid encountering potential threats in their environment. But like humans, adolescent mice seem to tolerate a lot of risk. So they spend a lot of time exploring a new environment and much less time than adults avoiding threats. And my lab has linked these behavioral changes to specific developmental changes in the prefrontal cortex. So our work shows how the developing prefrontal cortex actually adaptively supports adolescent milestones. And it doesn't simply remain in a suboptimal state as it's marching towards adulthood. So studying the neurobiology of threat avoidance is particularly important because excessive or inappropriate avoidance is a hallmark of a number of psychiatric disorders, including anxiety-like disorders and depression. So anxiety has a median age of onset of 11 years old. And early life stress is one of the major risk factors for developing these mental health disorders. So this is another major focus of my lab, is to understand how early life stress affects prefrontal development and the behaviors that rely on it. So most studies so far have focused on how a history of early life stress affects the adult brain, but we don't know the trajectory that leads there. So we've been studying this in my lab. We find that mice that exhibit or experienced early life stress exhibit much higher levels of threat avoidance already in adolescence. So they're acting sort of more like adults. And now we're investigating the underlying mechanisms using methods that allow us to monitor and manipulate neural activity in the developing prefrontal cortex while these animals are engaging in these threat response behaviors. So we think understanding how early life stress affects prefrontal development will be critical for developing early interventions for some psychiatric disorders. So that's the summary of what we're working on, and I'll pass things off to Anne.
Thanks a lot, Laura, and thanks so much to the host, and especially Coretta Harris, for hosting us today. It's really a great pleasure to be here, and thank you so much to all the audience members for being here as well. I got interested in neuroscience originally through a passion for cognitive development in children. And having worked a lot with elementary school kids as a school volunteer and a camp counselor, I was intrigued to understand how brains developed and how cognitive skills emerged. And I had an opportunity to do undergraduate research at MIT during this time when I was an undergraduate, and actually led a study on infant behavior, hoping to understand how infants' underdeveloped prefrontal cortex, like Laura told you about a moment ago, led them to make certain kinds of mistakes, especially in visual recognition tasks. It was fascinating work, and I was really inspired by other work in infants showing that rather sophisticated cognitive abilities, especially things like memory and a knowledge of natural physics, sometimes don't shine through in infants and very young children because of other limitations, like their motor system, which will turn out to be a theme in my research, and also in their weak cognitive control, mostly due to prefrontal cortex still developing, as you know from Laura's talk. So this was really exciting research, but I wanted to have the chance to measure neural activity a little bit more directly. So this is really hard to do in humans, and especially in human infants, because we don't have access to the brain, and certainly not easy access to the brain, and certainly not to single neurons, except during very unusual circumstances. So when I heard about an opportunity to study single neuron activity in non-human primates at UCSF, I thought this sounded great. So I headed out to San Francisco and began working in a lab that eventually led to my PhD, where I was studying how the brain processes motion in the world and uses motion signals to tell the body what to do in response. I was studying eye movements at this time, which is a pretty natural thing to do if you're studying visual motion. And the eye movement system is a great system to study because there are only 3 sets of muscles that move the eyes around. This was an amazing time in my life, and I really enjoyed collecting the data and analyzing it and discussing it with my colleagues. But when I was done with the PhD, I really wanted to return to the cognitive questions that were so interesting to me as an undergraduate. So I decided to join a lab that studied visual decision-making, which is what my lab works on now, and to try to use theoretical models to understand why we make the decisions that we make and why it takes the time that it takes to make those decisions. So both the speed and the accuracy of decisions was something that we wanted to understand. And at the time, I was focused again on visual motion. But when it was time for me to start my own lab, I wanted to take these skills and understand not just decisions about motion, but decisions that combine different sensory modalities together, which is, of course, something that we do all the time. Right? We might combine vision and hearing, for instance, when we're talking to someone. And when one of those signals is degraded, for instance, when you're talking on your phone and don't have the visual signal and can't hear very well, you know that it's really much harder to process that incoming language information, especially if you're somebody who's a little bit hard of hearing. And as humans, we very naturally put visual and auditory information together to have a better understanding of what's happening in the world. So I decided I was really interested in this process, how multisensory information gets put together. And I decided that I wanted to focus on vision and hearing, partly because of their relevance to processing language. But I decided that I wanted to study these kinds of decisions in rats and mice. And at the time, this was a really controversial decision among people who knew me, my postdoc advisor, and so on, because folks thought it was perhaps a little crazy to think you could learn something relevant to human decision making by studying a rat, right? After all, humans are really sophisticated and rats are pretty simple. So the first study that I did was actually to put rats and humans head to head. I did a behavior where I found a behavior that was developed in humans, and I found a way to bring it to rats. And I studied the same behavior. It was a multisensory behavior where the rats and the humans combined information from the vision and hearing and put it together to make a judgment about a flickering or a series of clicks. And then I just compared their behavior, and I compared their ability to optimally combine information from those 2 different sensory modalities. And it might surprise you to know that actually the rats and the humans were very similar. And I hope no one is disappointed to hear that. And this data really suggested that decision-making strategies can be common, and even the computations they rely on can be common across very different species. So to put it another way, rodents might be a lot smarter than we think. That could definitely be a problem if you're trying to get rid of one in your basement. But more to the point, having discovered these commonalities between rodents and humans, it put us in a great position because the tools that we have for measuring neural activity in rodents are super powerful and can let us make major strides in understanding how neural circuits really work. So to be able to have a behavior that was similar in both humans and rats was a great discovery. So we measured neural activity during these decisions in the rats, and we learned a lot about the neural circuits and how they put different pieces of information together, like from audition and vision. And we also learned something really surprising, which we did not expect at all. So even when our mice and rats were trained experts on a decision-making task, they moved a lot. And these they were often kind of fidgety movements we never trained them to make. And we found that these movements completely dominated the neural activity in their brains. So even though we were expecting to find signals that were related to the decisions that we were interested in, we found that most of what the brain was doing was responding to the movements that the animals were making. And I was at first really puzzled by this and started wondering, why were the mice moving all the time? Was it something about our experiment? And then maybe one day I had kind of an aha moment. I was on the subway in New York City, and when I looked around, I realized that humans are actually often moving all the time too, especially kids. They dislike sitting still and are making movements constantly. And this is no surprise to any of you who are parents. And for me as a parent, I'd also noticed that my kids moved a lot. Chair rocking, leg shaking, pen clicking, all movements that don't seem to be obviously related to the task at hand, and yet they're movements that humans seem to really want to engage in. And I don't totally have the answer for why that is today, but our observations, I think, suggest that maybe movements and cognition are much more linked than we previously realized. And that maybe one day we'll learn something really interesting about the brains of humans, perhaps especially human children, when we understand that link between cognition and movements a little bit better. Extending this approach even further across the lifespan to include mice at many different age points, including later in life, is something that we're working on right now, and is something that I hope to be able to tell you more about soon. We're still working on this problem at many stages of the lifespan, both in healthy adults, in healthy older adult animals, and in some younger animals as well. I feel really lucky to be at a place like UCLA where we're able to make the key measurements that we need in order to get to the bottom of this question. So thanks very much, and I will hand the microphone off to my colleague, Yirong Peng.
Good evening, everyone. It's my great pleasure to be part of this Women and Philosophy events. Thank you for the organizers and for every audience to participate in these events. So I'm talking about a little bit my background and talk about my research laboratory at the UCLA. So I was born and raised in China. I gained a strong interest in neuroscience when I did my undergraduate research at the Institute of Neuroscience, Chinese Academic Sciences. My research at that time was to look at what causes opiate addiction at the neuronal level. I examined what happens to a specific type of neurons when given morphines repetitively. So what I saw was a very dramatic decrease in the number of opiate receptors in the membrane of these neurons. That means the chronic exposure to opiates desensitizes the neuron's ability to respond to the drug, and the neurons require even greater dose to produce the same effect. From that experience, I learned that neuron has great plasticity to adapt to their environment. Some adaptations are beneficial, but some, like addictions, are detrimental. So inspired by this summer of research, I joined the same institute for my graduate study to understand neuronal plasticity. As I learned more about the nervous system, I become more and more intrigued by its complexity, and I'm curious to know how the nervous system is structured so we can perceive the world around us. I decided to study the development of the nervous system during my postdoc training and in my own lab. My simple idea is that if we can figure out how the brain is formed, and we may know how to repair it once the degeneration happens. So currently, my research laboratory at UCLA focuses on understanding how the visual system allowed us to see the world with the great resolutions, and what happens when, whether by diseases or by agents, visions become impaired. So as we all know, vision begins in the retina. The retina is a neural structure at the back of the eye. One fascinating thing about the retina is that it's actually a part of the brain. That's because it shares the same embryonic origin as the rest of the brain, and because it's directly connected to it. So when light hits the retina, there are multiple neurons that transform light energy to electric signals, and that transmitted to the brain. So the brain then processes these electrical signals into images, thereby creating visions. So the structure of the retina is complex and beautiful. So in my virtual backgrounds, that's actually an image of the retina under the microscope. So there are over 100 neuron types with different shape. Any defects in the shape of the retina cell could cause visual defects. So my laboratory is working to catalog and understand why there are so many different shapes, and why some of them are susceptible to generations which can impair vision. To address these questions, we think the key is to understand the complexity of the overall structure. We aim to generate a roadmap in which we will know which genes are responsible for every neural variations. With that kind of knowledge, we should also be able to identify which gene can cause the degeneration of specific type of neurons. So we are currently using a multidisciplinary approach to figure out the patterns in family of neurons that are similar to each other and may share a common gene responsible for their characteristics. With this approach, we have successfully identified genetic regulators that control the creations of specific neuronal shape. So in order to translate the basic discovery to a potential therapeutic strategies, we also use an in vitro model in which we can grow a retina-like structure from human pluripotent stem cells in a petri dish. This structure we call it a retinal organoid, or call it a mini retina. And we can genetically manipulate the organoids to grow specific cell type. So the hope behind this line of research is that this approach will eventually be used to grow cells to replace the degenerate cell in order to rescue vision. So in conclusion, vision is a gift, but unfortunately losing vision is a frightening prospect. Our research aims to lay a foundation to understand human vision and find innovative strategy to rescue or prevent vision loss. That's a very brief summary about the research conducted in my lab. I'm happy to address any questions during the Q&A. Now I will give the floor to Jessica.
Hello, and thank you everybody for participating in your interest in our work at UCLA and in women neuroscientists. And I'm so impressed by all of my colleagues, and I've been enjoying all of their presentations. So they're studying basic mechanisms of disease, of the nervous system in the brain, and how that impacts development and behavior in such elegant ways. My lab is studying something complementary to that, which is how the brain degenerates in disease. And I'm going to tell you a little bit about how I got started and came to the questions that we're asking every day. So, you know, as many people who ultimately come into medical scientists and scientists, I was touched by a experience as a child seeing a family member with Alzheimer's disease. And I was very thrown by what it means to lose your memory and lose your personality and yourself while you're still alive and otherwise well and moving through the world. And this was very disruptive to me, and I thought it was very unfair. And interestingly, at the time, we didn't know, and we still only know so much about neurodegenerative disease. And so I remember thinking about this problem as like a giant black box, really where we only really had a couple ideas about maybe what was going on. And I went to my undergrad years, kind of not quite sure what I wanted to do. Professionally, I was very interested in learning things. And I, towards the end of my undergrad, learned about this career as a physician scientist, where you both get trained as a physician and you get trained as a scientist. And you kind of have the role of bridging medicine and science. And so that felt right, because I saw myself as somebody who was constantly learning and connecting the dots across the different classes and disciplines that I was studying in. So I thought this was going to be a great field for me. So I started off on my training as a physician scientist at UCLA and Caltech, and wanted to learn in my first phase how one uses science to come into an unknown space of relevance. So I did my training at Caltech in biological chemistry, and the first paper that I wrote was inventing a new method to visualize this protein signal that I thought was going to be relevant, and many others thought was going to be relevant for human disease, but we didn't really have a way of seeing effectively at that time. And so that opened up a lot of opportunities to study the basic mechanisms of that process and learn completely new things that we didn't expect to find. And that experience was very supportive of the power of science to open up new areas in the scientific method to provide like a rigorous framework for how to attack the unknowns. But I felt that I needed more training to learn how to apply all of these new methods in science to medical problems. So after I went back to medical school and finished medical school, and actually training in neuroscience, in neurology, I went back to the lab as a postdoc, learning functional genomics and systems biology so that I would have ways of leveraging human data to identify those unknown targets that I could then employ the mechanism tools in science to study and attack. So specifically, my lab now focuses on trying to figure out what drives neurodegeneration, which we can very humbly say we still don't understand what really causes Alzheimer's disease and related diseases. So we're trying to employ large data techniques, specifically interested in what drives the neuroimmune and neuroinflammatory aspect of disease. And why am I doing that? Well, I want to find therapeutic avenues out of human data. And it takes a long time to do that. But when you get to that state where you're pushing something forward, you know that that observation came directly out of human brain data. And I'm very excited about the fact that we have the technology to do that these days. And so we're pushing forward on that path. And the lab is currently engaged in a number of studies that are looking at individual cells in different dementia brains and different brain regions in different stages and characterizing what they look like, what's driving their signaling, and how they relate to the genetic factors that influence our differential risk of those diseases. And so from that work, we have, some insights perhaps, or inspiration that is shared by some of the audience members who put in some questions. So one of the questions that was asked earlier was, what are the neurological factors in aging that have been identified by our research? It's very important to recognize that the shared risk factor of most of of many adult onset neurodegenerative diseases is age. We go for decades without these diseases, and then suddenly a certain age, the risk goes up and up and up. So age must be a factor. And one of the things that we've found is that, as the brain ages, how we respond to DNA damage is one potentially really important factor that could be a convergent point between aging and neurodegenerative disease. And the person-to-person variation in our genes can influence this. So learning precisely how age-related damage to cells leads to toxicity is an important fundamental question that I think answering is going to allow us in the field, to influence many different age-related brain diseases simultaneously. And another really important area of interest in my lab in particular is the aging of the immune system, and how that impacts the brain is an area where we have a lot of unknowns in the field, but the tools and the opportunities to dive into this area are all in hand, and we are seeing a real accelerated understanding of immune system roles in the brain, including things like how it's modified by aging. When you think about this, one of the things that my data informs is a suggestion that the immune system might not just be simply too much or too little, but might be more of a matter of maybe too little of the wrong things and too much of the wrong things. So there's some specificity in how the immune system is being dysregulated in disease, and that might also change over time. So one of the ways that we think about that is when you age, your immune system loses the ability to recognize damaged cells or viruses as well as a younger brain or younger immune system would be able to. And our modeling suggests that it's possible that in a similar way as our immune systems age, they may not clear some of the early toxic species that ultimately may drive neurodegenerative disease if they accumulate. So this is one example of where our modeling converges with this kind of framework of thinking, and we have very specific targets and drivers in hand now to study those mechanisms in different ways, including like Yirong in iPSC models, and also in using human data, for example, with the incredible human precision health data set that's been curated here at UCLA. So I want to turn over to the next speaker, Dr. Aparna Bhaduri, for her presentation. Thank you.
Thank you, everyone, for this amazing event, and thank you to Women in Science in Neuroscience and Philanthropy for organizing, and thank you to those who are here for your interest in the brain. Unlike some of the other speakers, I took a little bit of a long way to get to neuroscience and studying the brain. I was always interested in science and research and was always an enthusiastic participant in science fairs as a child and was able to do research as an undergraduate at Rice University in Houston. But I was also interested in being on the debate team and majoring in political science. So it took me a little while to decide that I wanted to pursue a career in science. Ultimately, I did decide to do a PhD in cancer biology at Stanford University, where I studied both skin cancer and skin development. And it was there that I really started to appreciate that cancer and development are kind of two sides of the same coin. Upon graduating, I knew that I had always had a long-term fascination with the human brain. I still think it's the most beautiful thing that evolution has ever done, is creating the human brain specifically. And so I really wanted to be able to study that. Luckily, I was able to transfer some of my skills at looking at big data sets to my postdoc at the University of California, San Francisco, where I worked with Dr. Arnold Kriegstein as part of a brain initiative, which is a federally funded program to understand cells in the brain. And we specifically started trying to identify what are the various cells that actually exist in the brain. It might be surprising, but scientists still don't know what all of the cells are and what they look like and what they do in the brain. And we think that there are thousands of cell types in the brain. I started my lab at UCLA almost 2 years ago in 2021, and we have been studying both normal brain development as well as brain cancer. And in my lab, we use models of the developing human brain because the human brain, although it shares many characteristics with other model systems, including rodents, also has very unique features. And specifically, we know that the human brain is larger than other brains, and that this process of expanding both to new cell types as well as to expanded areas of certain parts of the brain happens during development. And so we're able to use something called organoids, which are these stem cell derived models of the brain, to really look at some of the processes that happen during these developmental stages. And one of the things that we're really trying to understand is how do the stem cells of the brain make this huge diversity of cell types that give us the ability to engage in these cognitive functions and these other processing functions that we as humans do on a daily basis. Now, I mentioned that cancer and development are two sides of the same coin. And one thing that's been really fascinating for my work is our ability to use cancer, specifically glioblastoma, which is the most common and most aggressive form of adult brain cancer, to compare and contrast between the different states. And one thing that we see is that there are stem cells that exist during development that are thought to disappear after birth, that come back and seem to be aggressive drivers of tumor formation and progression. Now, one of the things that's really interesting about this is some of those stem cell populations that exist in the cancer don't exist in rodent systems, or at least not very frequently, even during development, which means that if we want to study the human aspects of this cancer and why it is so devastating to individuals, we need to be able to study human systems. So one of the things that my lab has been working and developing is what you see pictured behind me, which is a picture of an organoid, which we see under the microscope. And in the green cells are tumor cells that have come directly from a patient from surgeries here at UCLA. And the cells have been transplanted into our model of the brain that we have in a dish. And this allows us to study cancer progression. So through this work and through these systems, we're able to look at not only normal development, but also how cancers develop and really use what we learn from one to inform the other. And we hope that this will allow us to develop treatments for these cancers and to better understand how they work, as well as to cross-inform our understanding of how we really become human. So with that, I'd like to turn it back over to Dr. Marcus as our host and moderator, and I'm happy to take questions.
Thanks, all of you. Wow, that was such an inspiring, I'm a neuroscientist and I was inspired and I learned so much. So it was really a fantastic set of talks. Some of you have touched on this, but those of you who maybe didn't, Is there something you're particularly excited about having an impact through the study, through neuroscience research, having an impact on human health? And I'm going to say in our lifetimes, although I probably should say in your lifetimes, because you all are younger than me, where you really, see the potential that we're going to be able to take research out of the lab into, patient care. Is there anything that you're particularly excited about, whether it's your own research or something else that you've seen, or a technique that you'd like to share with the audience? Anybody who would like to start? Okay, Jessica, please.
So yeah, so in my kind of physician scientist role, I'm always thinking about that problem of how, we have, when you're in your science life, you see this incredible progress. You see breakthroughs that you never would have imagined. And then you go back to your doctor life and you see all of these patients that you still can't do bump kiss for. So I think about this a lot. And I think that from that perspective, I can say that everybody on this panel is doing very relevant work with a promise of informing, human care, whether it's, treating a disease like glioblastoma or Alzheimer's disease or macular degeneration, or whether it's understanding human brain development and behavior and, how to work with adolescents and psychiatric disorders as well. So, and the reason why is that we have the opportunity to do things with data that we never had before, where the incredible detailed analyses that can be done experimentally can actually be, in a data way, transferred over to what's going on in human, where we have access to have longitudinal and observational data that we never had before. So all of these breakthroughs that are happening at the bench can be directly compared to very, very detailed human data. And that's going to just accelerate in a huge way the ability to translate. I know a lot of experiments that I did at the bench as a graduate student were completely contained to that experiment, that Western blot, and they never were compared across other data sets from human or other scientists' hands. And we do that now, and that's going to be routine in science in the future. The second thing is the iPSC modeling as a complementary model to go with things like the animal models that have the full organismal platform, because we can insert into that kind of comparison of data sets a cellular model that's completely human, and take a giant missing link that we had for a really, really long time to that translation and finally start to apply it. So we're in an incredible time for being able to accelerate the translation of discoveries from the bench to the bedside.
That's great news for all of us to hear. And it prompts me to ask Aparna and Yirong, since you both talked about using, IPS mediated model organoids, retinal organoids or brain organoids, where are we currently and how closely those organoids in a dish, those little mini retina in a dish or mini brains in a dish, how close are they to actually representing the human retina or human brain? Either one of you want to speak to that a bit?
Sure, I can start with the brain and then I'll hand it off to you, Yirong, for the retina. But I think that one thing that's important to remember about these models is that they really are simplified models of the brain. So they're reductionist models, and they're ways for us to ask questions in a dish, which is really exciting because we can do this in human cells. We can do it on a number of different cell types. So we can look at the stem cell and the cells that it makes, and we can do it with human tumors. But the organoid itself is not very close to a brain. It's much smaller. It's missing a lot of different parts. We usually look at one brain structure at a time. So for example, in my lab, we focus on the cortex, but there's other really important structures, including the thalamus and hypothalamus and cerebellum and other things that all need to connect together to really make a functional brain. And we only have some of the cell types that exist. So we have the ones that come from this particular stem cell population, but we're missing a lot of cell types. So even though that it's a really great system, there's also important limitations to it, which is why research in this area is incredibly exciting and should be paired with the other models that are used, for example, the ones that Laura and Anne are using to be able to really integrate cellular things that we see at the cellular level in those experiments with what's actually happening in terms of behavior and decision making, etc.
Yeah, I totally agree. I just want to add a little bit more on my insight when I using the retinal organoids, really looking at how close the organoids seem to the human retina. So why we want to use the human model or mimic the human model, because there's just dramatically species difference when we're looking at even the anatomy structures. For example, especially for visions, we may think, okay, the retina is very conserved among all the species. But when we're looking at how we perceive the world compared to other species, they may be different. We may not see the greatest speed. We cannot detect fast moving thing. We may not detect the contrast sensitivity. But what we can do is we can see everything clear. That's why we can read in, because that's kind of a very small visual difference on your visual world. So that's all coming from the retina, because our vision especially is a central vision. And that's because we have a very specialized retina structure in the center part of the retina. And this part is largely missing for most of the mammalian species. So that's why the retinal organoids become very feasible or highly potential to really mimic the human retina. But the question is, the human retina organoids is a very simplified structure. It lacks the regional specialization. But now, combined with the big data research, we know what specific gene is especially expressed in the central areas. Then we can leverage that gene information to see how we can create a retinal organoids most mimic to that central part of the human retina. So why is it important? Because for the vision defects, especially for the Americans, the leading cause of the visual loss is coming from the age-related macular degeneration. But unfortunately, the macula is actually the central part of the retina. So if we don't have a good model to mimic that structure, we may never get the step to figure out a cure to rescue that visual loss. So it's really important to start using the retinal organoids and to really think about how to make it very similar to the human retina.
Oh, thank you. That's great. I'm going to ask one last question of the panel and then turn it over to questions from the audience. So if people in the audience have questions, you can enter them in the Q&A. But all of you talked a little bit about how you sort of got interested in the field, sort of the early beginnings of your becoming a neuroscientist. And it makes me think to the trainees now, the trainees that are in your labs now, that will be sort of the future of the field, that will, and, you know, how do you, what do you have any observations about how trainees in neuroscience are doing these days? Are they, particularly at UCLA, or something to speak about the trainees in your lab that you'd like to talk about?
Sure, Laura, do you want to go first and I'll follow you?
I'll go after you. Go ahead.
Okay, yeah, I think it's one of the things that's great about UCLA, and one of the reasons that I came here is that the students and postdocs here are fantastic, and they have incredible enthusiasm for neuroscience and come from really strong backgrounds. I think one of the things that's been an interesting development in recent years is the growing enthusiasm that the students and postdocs have for learning more quantitative methods. They've really started to realize, kind of dovetailing on what Jessica mentioned a moment ago about really large data sets. That's so true. And now we need new methods to analyze these data sets that are really different from what we used to use, even back when I was a student or a postdoc. And the new trainees recognize that, and they are really, really eager to learn as much as they can in terms of quantitative analysis, so that they can really take their experiments to the next level. So that's been really exciting for me to see, because I have a lot of enthusiasm for that approach as well. I think our biggest challenge here is making sure that we have, could provide them all of the tools that they need to learn these new skills.
Mm-hmm. Great. Thanks. Laura.
Yeah, I agree with everything Anne says. I've been so impressed with how my trainees have been getting the new skills to analyze these large data sets we can collect. So there's all of these computational academies that you can attend, and they've been doing that. So that's been great to see. And I've also just been really impressed with how collaborative all my trainees are with each other. It's been really lovely to see them working together. I have a lot of PhD students, and they're making so much progress working on these collaborative projects. And I think this is really important these days. When to publish your research, you have to do so many experiments to get a paper published in a journal. And so these collaborative efforts are really a win-win situation for the students. And I've also been impressed with sort of their bravery in tackling really challenging problems. So when you study the developing brain in freely moving animals like we do, it requires us to implant these tiny little optic fibers into the brain so that we can record the neural activity as they're moving around and turn neurons on and off so we can understand their causal roles and behavior. And you talk to, older people who have been in the field for a long time, like, oh, you can't do that. But I'm like, no, my grad student, she just went for it. She was really resilient to the failure and got these really challenging experiments to work. So I've been very impressed with their resilience and sort of bravery.
Fantastic. That's fantastic. So we are getting some questions from the audience. So I'd like to turn to that now. The first one actually, Laura, is for you as to whether you found any ways to reverse or counteract overactive threat perceptions, or even how you might find ways to do that.
That's actually a nice segue from what I was just talking about. So this is what we're working on right now. So, so the prefrontal cortex acts as sort of a high order center that it sends axons, which are the wires of the neurons, and they connect to many different brain regions. So if this is prefrontal cortex, it's sending axons to a lot of other structures that can tell an animal to approach something or avoid something. In development, we're not sure what signals are actually being sent out by the prefrontal cortex. So that's something really important to understand. And I don't have the answer yet, but these experiments are ongoing. So we can implant these little fibers into the brain and see what signals are happening in the prefrontal axons in very distant brain regions. And that will allow us to understand if the signals are aberrant, and if so, what specific pathways are affected. And then we can go in and try to use either sort of genetic and molecular approaches to rescue that, or sort of behavioral therapy type things to rescue that.
Wow, fantastic. That's awesome. So a question that's sort of for anyone who wants to answer, although probably a little more relevant for some of you than others, but are the issues that you're addressing and the discoveries that you're making different for men and women, or are they consistent for both? Is anyone looking at issues of, sex differences, or observing any issues of sex differences?
I can speak to that a little bit. So one of the things that we look at in my lab are the neuroimmune and neuroglial interactions in Alzheimer's and other dementias. And, we have a big data set where we are describing all the different cell types and how they change in disease. And that data set does support some differences between men and women that are statistically significant, that reproduce actually some observations seen by other people's work. So that when you start to see this reproducible findings, then you get a little bit more excited. And those observations center around there being maybe just a little bit of a different neuroinflammatory profile in Alzheimer's disease brains, in microglia, in men and women. And my data is what we call confounded by a variable that I can't tell you how much it may be impacting the difference we observe, because it's not equally split between the groups. And that's a difference in the ApoE genotype, which also can influence microglia. So I have to have a little reservation in what I say because of that. But in mouse studies where there's been control over that ApoE gene specifically, there's also been some suggestion that the way it impacts microglia neuroinflammation may have some sex-dependent features. And so I think that there's going to be something there. How much it impacts function or disease, we don't know, but it does seem to be a theme that's coming out of multiple different types of data right now.
That's great. Thank you. I'm going to squeeze in one more question, just because I think there might be a lot of members of our audience that would be interested in it. And we've learned about, adverse child events and the impact on brain development and aging and neurodegeneration. are there anything, lifestyle things that you can do, to live longer, healthier lives, to, delay, cognitive impairment with age? Any thoughts about that for the audience to take away? Who'd like to go first?
As a
Okay.
neurologist who sees patients in the dementia clinic, I feel like I have maybe a role in this. So a lot of dementia in this country is actually due to not pure Alzheimer's pathology, but actually in component of metabolic disease and cardiovascular disease. And so that is probably the lowest hanging fruit as a modifiable risk factor. And you see this being borne out in studies right now, in populational studies, that things like making sure your blood pressure is under control if you have high blood pressure, or that your diabetes is treated, or that you have a healthy, lifestyle, that those things make a difference. And that's because of that. So there's when you look at the population, there's a huge potential to reduce the number of individuals with dementia by taking care of those simple things, like just having a healthy lifestyle and taking care of your underlying medical problems and risk factors.
All right. Anybody else want to weigh in on that, Anne? Yep.
Yeah, I'll add one sort of related perspective, because my lab has been focused not on neurodegeneration, but on healthy aging, and trying to understand how the brain changes during healthy aging when there is not disease present, which does happen, which does happen sometimes. A lot of people get Alzheimer's, but a lot of people manage not to. So we've been trying to understand this. There's a lot of open questions. We were inspired a lot by a really interesting study of superagers that was done at UC Irvine by Claudia Kawas and Maria Corrado. And they studied people who were in their 90s and tracked them over a number of years to try to understand what happened. And sort of features that were protective. And they found that it was a really idiosyncratic set of features that was associated with healthy aging. I think it's important to take them with a grain of salt, because there might be a feature that's correlated with healthy aging, but doesn't actually doesn't play a causal role in protecting longevity and also brain health. But a couple of interesting things that they found for people who were in their 90s that were healthy agers, they tended to be of normal weight or slightly overweight. So if you're in your 70s and want to live 20 years longer, go have dessert tonight. It might help you. They also had moderate caffeine intake, and a really important feature, one of the only ones that was important both for aging and for protection against dementia, was light physical activity. So people who either were not active or people who were active but unfortunately had to stop due to an injury were at greater risk of death and also of dementia. So I thought that was an interesting factor. It didn't have to be any kind of crazy exercise, but just some kind of light exercise in that superager study was very protective both for longevity and for dementia as well.
All right. Thank you all so much. We are coming to the end of our time, and I think the go have dessert recommendation is a good way to wrap up us talk about neuroscience research. I want to thank all of our panelists. You guys are amazing. The research that you do is amazing. Thank you for doing it. Thank you for dedicating your lives to helping children, adults, to help us understand the brain and how it develops and how it ages. I'd like to thank the Women in Philanthropy for hosting this event and for bringing together such amazing scientists and researchers to share their work. And I want to thank the audience for your participation, and as others have said, for your interest both in the brain and brain science and in UCLA and the women here at UCLA that are doing such amazing things. So with that, I'm going to turn it over back to Dr. Harris to close out the session. Thank you all very much.
I will just say, wow. A heartfelt thank you to Dr. Emily Marcus, Dr. Aparna Bhaduri, Dr. Ann Churchland, Dr. Laura DeNardo, Dr. Yirong Peng, and Dr. Jessica Rexach, for your time and enlightening us on how our brains work and what goes wrong with our brains when our brains do not function properly. I'll also say to Dr. Marcus, thank you for making me an honorary doctor during this session, as I am not a doctor, but thank you for making me one during this session. Thank you all. Thank you as well to Women in Philanthropy, members and other UCLA supporters for participating in our webinar. And also tremendous appreciation to Juan Mejia from the Development Events team for helping us with all the Zoom meeting logistics. We will be hosting webinars and in-person programs after the new year, so look for invitations to other interesting programs from Women in Philanthropy. And with that, I'd like to say good evening and thank you for attending.